Thank you so much to all of our presenters for your great information and taking the time to share it with everyone. So for the audience, if you have a question you'd like our presenters to answer, please enter it into the chat pod at the right. And with that, we're going to go ahead and get started with Dr. Getie-Kebtie. So specificity, this is for Dr. Getie-Kebtie. Specificity and selectivity always confuse us. Usually we only perform selectivity validation runs. Under which circumstances should we perform a separate specificity run?
Hi, can you hear me? Hello?
Yes, I can hear you loud and clear. Very good. Thank
you. Yeah. Okay. Yeah. So it's a very good question. I know that this has been asked in the morning with respect to PK assays, and the definition of selectivity and specificity as applies to immunogenicity is more or less similar to the PK assay. So when we speak of selectivity, the reason why we perform selectivity is to confirm the ability of the assay to identify ADA specific to the therapeutic protein in the presence of other sample components. And this is usually performed using 10 independent negative control matrix samples spiked with the low and high positive control sample concentration. Now, with respect to specificity, this is performed to confirm the ability of the assay to exclusively detect the target ADA, but not other molecules which may be present in the sample. And this is also usually performed using 10 independent negative control matrix samples in the absence of the positive control. And thank you.
Great. Okay, thank you so much. Another question for you. Is incurred sample reanalysis required for immunogenicity testing? If yes, what are the conditions we need to consider?
Yeah, so for immunogenicity testing, at the moment, incurred sample reanalysis is not required as it is for PK assays. And so the answer, in short, is yeah, we don't require ISR for immunogenicity assays.
Great, thank you so much, Doctor, for your answers. All right, our next question is for Dr. Cai. How would you differentiate prodrug delivery systems to liposomic delivery systems?
Hi, can you hear me?
Yep, we can hear you loud and clear. Thank you.
Great. So liposomes, the liposome delivery system is quite different from the prodrug delivery system. Because liposomes have the lipid bilayers with aqueous phase to encapsulate the drug compound. So with this design, you enable a diverse range of drugs to be encapsulated and then enhance targeting and reduce toxicity. For the prodrugs, they are derivatives of the active drug moieties. So with the modified chemical structures, they are designed to enhance ADMET.
Okay, great. Thank you so much. We have one more question for you. And if you can speak a little bit closer to your mic or a little louder, I think that'd be great. So, all right. Question for you. For an oral drug product that disintegrates quickly upon administration, for example, no plasma concentration after 5 minutes, are evaluations ex vivo conversion necessary? What kind of testing do you recommend? What kind of testing do you recommend, or what would the FDA require?
This is a great question. So for prodrugs that disintegrated fast upon administration, in the case that when no plasma concentration of prodrug after 5 minutes in that case, it may not be feasible to quantify the prodrug in the plasma samples. And if there is no prodrug left in the collected blood ex vivo, then the evaluation on ex vivo conversion will not be necessary, in my opinion. However, regarding to the study design and what type of analyte should be measured from the clinical study, this should be something I think they should consult with the FDA, the Drug Review Division.
All right, great. Great answers. Thank you so much. Our next question is for Dr. Zhang. What is the acceptance criteria with regards to temperature and humidity of DBS blood samples during storage and transportation?
That is a very good question, and I don't think there's any set acceptance criteria. Everything is depending on the design of the study of the drugs and also the validated method parameters. It's really hard to say one for all. So the sponsors have to determine what's the best for their drugs and their study.
Great, thank you so much. We have another question for you. How does the HCT level affect analyte quantitation?
So basically, the HCT level determines the viscosity of the blood, therefore have an impact on the spreading of the blood on the collection cards. Basically, the blood with higher HCT, they will spread less compared to the blood with lower HCT. So if they were punched, use the same size of the puncture, the volume may be different. And if the calibration curve is established in blood with, let's say, intermediate HCT level, it could result in underestimation of the DBS with lower HCT or overestimation in the case of DBS with higher HCT. Additionally, it has been observed that the different HCT level may have an impact on the extraction efficiency. So consequently, the recovery would be also impacted. So in that way, the measurement of the drug concentration from the DBS could be impacted as well. So that's why it's important to determine if the HCT variability could be affect the analyte quantitation.
Great, thank you. And we do have one more question for you. Can DBS variability be reduced by taking multiple punches for a single sample point?
This is also a very good question. I think the answer is depends. So basically, if the single sample point is homogenic by itself, I think it might reduce the variability. However, sometimes there's not a choice because the samples were taken, not homogeneously. So it actually could introduce more variabilities. Depends on different locations where the punch is taken from that single sample point. So that actually goes back to my talk. That's actually another good reason to always validate those parameters. For example, choose different positions to punch for a single sample point and to see if there's any impact.
All right, thank you so much, Dr. Zhang. And now we have questions for Dr. Ayala. So the question is, were the methods using similar numbers of runs in your presentation? Also, even though the samples were lower than the HQC, how do you justify accepting data between the MQC level and the unreliable HQC?
Thank you for the question. There were some differences in the numbers of runs among the methods. I redid the numbers right now, and the method number 1 still has some of the highest failed runs among the methods. The second part of the question is related to the reliability of the concentrations measured in subject samples. The method included 4 QC levels, if I recall, and so most of the subject samples fell between the two MQC levels and then the LQC level. So we had at least 3 QC points bracketing the subject sample concentration. So in my opinion, I didn't really have much concern with the reliability of subject sample concentrations.
All right, great, thank you. Another question for you. For the histogram comparing between methods, would it be possible that we tend to see more issues for the method more frequently published and tend to see less issues of the method people rarely use?
So the way I interpreted this question is, do we see more issues with methods that are being used more frequently versus a method that's not used as frequently? In fact, that wasn't the case in this particular analysis. Method number 1 was used for the very first time, and it had an increased number of issues among all the other methods. And so I would say no. You do see increased, at least in this analysis, increased issues with less frequently used methods.
All right, great, thank you. And we have a question for now Dr. Davila. So Dr. Davila, my CRO was just recently inspected by FDA for BA/BE studies. When can we expect another inspection?
Yeah, so FDA aims to conduct clinical and analytical site inspections of CROs, conducting the clinical and analytical portions of BA/BE studies every 2 to 3 years. However, the agency may initiate an inspection before that time.
All right, thank you, Dr. Davila. All right, we have another question for Dr. Getie-Kebtie. Whether Cmax or C-trough drug level, which one should be considered to determine free drug in an immunogenicity assay?
I'm trying to unmute myself. I think I'm unmuted, right?
Yep, we can.
Yeah, so can you repeat the question, please?
Sure. All right. Whether, and hopefully this is clear to you, whether Cmax or C-trough drug level, which one should be considered to determine free drug in an immunogenicity assay?
Okay. Yeah, so generally, so when you are assessing for drug tolerance in immunogenicity assay, you should consider your PK concentration in the sample that you are analyzing. And the Cmax is generally considered to be the one that you have to target for, but it also depends on at what time point you are drawing your samples, and your drug tolerance should consider for the amount of drug that could be present in your sample.
Wonderful. All right. We have several more questions for you. And our next one is, in recent years, laboratories performing immunogenicity assays have been given observations against the draft guidance and the current 2019 guidance regarding new expectations for LPC, cut point determination, and NAD selectivity. Even when reviewing study conduct that occurred before the draft guidance was issued and now finalized. What is FDA's position regarding this observation against guidance, which was not available at the time of assay development, validation, and method transfer?
Okay. So generally, the expectation is that if your study is conducted after the guidance is issued, you are expected to validate your method in accordance to the current expectations. And so if the validation was done several years back, then you have to make sure that the method is still applicable under the current expectations. And so what we really look for is when your study is conducted, not necessarily when your validation was performed.
Great, thank you. Great answer. All right, another question for you. If we do not include calibration curves in our assay, how do we determine precision? Is looking at variation, the percentage of CV between signals of the different wells sufficient? Or must we include curved, although immunogenicity is qualitative? What do we do if the signals are highly variable at the high range controls? This affects titer repeatability.
Great question. Yeah, so immunogenicity assays are not quantitative assays, so you don't need to have a calibration curve because you're not really determining actual ADA concentrations. Rather, you are determining whether your sample contains ADA or not. And so for precision, as it was indicated in the question, the assay signals are used for a determination of the precision of the assay.
Great, thank you. And we have one last question for you. Should MRD be included in reporting sensitivity?
Yeah, the short answer is yes. And so assay sensitivity should be reported after factoring in the MRD. For example, if an assay has a sensitivity of 10 nanogram per milliliter, I'm giving just an example, with MRD of 10, then your sensitivity will be 10 times 10, which is 100 nanogram per milliliter.
All right, thank you so much, Dr. Getie-Kebtie, Dr. Cai, Dr. Zhang, Dr. Ayala, and Dr. Davila for your presentations and for taking